Okay, guys, we are going to work on chapter 8 now. It's our nervous system. This is probably our largest, or not probably, but definitely the largest portion of our next unit. And the lecture seems really long. There's a lot of diagrams along the way to help us understand these concepts. So I'm going to break this into parts. This first section, we're going to focus on kind of some of the overview and main divisions and some of the physiology side of things. And then in the second half, we'll talk a little bit more kind of through just the anatomy of our entire nervous system. So our nervous system is divided into two main divisions. We have our central nervous system, or CNS, as well as our peripheral nervous system, or PNS. So our central nervous system is going to consist of the brain and spinal cord, and the PNS is basically going to be everything else. So the nerves that go to our face, the nerves to our upper limbs, the nerves to our lower limbs, all the nerves that run to our abdominal organs, all of those are going to be in some way considered part of the PNS. So our nervous system functions. Its job is to receive sensory input. So we sense information first. We sense it in the periphery first. We send that to the brain and spinal cord to integrate that information. We also control muscles and glands. It has a role in helping us maintain homeostasis or our balanced environment. And it also, of course, is important in establishing and maintaining mental activity. So we just said above, the central nervous system is our brain and spinal cord. The peripheral nervous system is all the tissue that falls outside of our central nervous system. And for both of these, we're going to talk about a sensory division. And so our sensory division is going to send signals to the CNS, the central nervous system. So that's from the periphery to central. So sometimes we call that afferent division, the afferent division. So afferent arrives. So think that's the information arriving to the brain. And then we have our motor division, or efferent division. Think efferent exits. So that's going from the brain or spinal cord back down the chain to either a muscle, an organ, a gland, whatever is the effector that's going to do their job. So we also can break it down into this kind of three different divisions of somatic nervous system. So somatic is going to mean pertaining to the body. So this is going to be our central nervous system to our skeletal muscle is our somatic nervous system. Autonomic sounds a lot like automatic, so that's kind of how you should think of the autonomic nervous system. It's going to be from our central nervous system to our involuntary structures. That includes things like our cardiac muscle, our smooth muscle, and our glands. And then this enteric nervous system we won't talk a lot about, but I just want to kind of make you aware of it. But we have a special division of our nervous system that's only in our digestive tract. And this actually functions completely independently of the central nervous system. So when you have a gut feeling, you can blame your enteric nervous system. So there is some communication coming between our guts and our brains. And this just kind of shows you that breakdown here, and we'll discuss this a little bit more going further. So for our nervous system, we have two main types of cells. We have the neurons themselves. These are going to be our nerves that receive stimuli, conduct our action potentials or nerve impulses, and how we transmit signals to other neurons or whatever the effector organ or tissue is. Our glial cells are more of our supportive cells. They're found in both the central and peripheral nervous system. They do not conduct action potentials, so these cells are not involved in the impulse transmission, and they have various functions. So we'll discuss the few different types that you'll need to know. So the neuron itself, in terms of the anatomy of a neuron, we have a cell body, which is also called a soma. This is going to be a single nuclei, not our multinucleated, like some of our muscle cells, but just a single nuclei. We have dendrites that are branches off of our cell body, and this is how our neuron is going to receive information from other neurons. And then our axon is typically just a single long process that's leaving the cell body and that's going to conduct signals to the CNS if they're sensory and away from the CNS if they're motor. So here's our neuron. This main portion is the cell body with our single nucleus. We have our dendrites off of the cell body, and these are going to be receiving signals. And we have our axon off of the cell body that's going to be sending or transmitting signals. We have a few different types of neurons. This multipolar neuron is probably the most common type. So we have multiple dendrites with one axon. A bipolar neuron just has one dendrite projection and one axon. And then we have a pseudo-unipolar neuron. This one we really don't talk about very often, but it has an axon that functions kind of like a dendrite. So it has a sensory portion of the axon, and this other half would be where it's sending its signal. All right, glial cells are our supportive cells. So you, like I said, you do need to know and be able to kind of match up the function with the different types of glial cells. Our astrocytes are major supporting cells. So their main job is to stimulate or inhibit activity of neurons that are forming what's known as the blood-brain barrier. So our brain obviously is very important. So we have this extra security measure called the blood-brain barrier to prevent substances that shouldn't be there from getting in. So it's our astrocytes' job to maintain that blood-brain barrier. Epidermal cells, these are found in a few different cavities in the brain, and they are responsible for helping to produce and move cerebrospinal fluid that we'll talk about in the second half of the lecture. Microglial cells are our immune cells in the central nervous system. So they're going to try to eat and remove bacteria and cell debris. Oligodendrocytes and Schwann cells perform more or less the same function, but they're found in the two separate divisions. So oligodendrocytes provide myelin to axons in the central nervous system. Schwann cells provide myelin to axons of neurons in the peripheral nervous system. And we'll talk about what myelin is here in just a second. So the myelin sheaths are specialized layers that wrap around the axons. They act as insulation for our axons. Like we said, this is done by two separate glial cells, oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system. And so, as I was saying, myelin's a great insulator. And so it's going to make sure that all the ions involved in our movement across the cell membrane that creates our action potential and nerve impulse are maintained. So you can think of our nerves kind of like our electrical wiring of the body. And so if you think of how a wire has a cover on it, that is to help it maintain all the electricity. So our myelin is helping us maintain that signal within the nerve. So a nerve that is myelinated is going to be able to transmit a signal much more quickly and efficiently than an unmyelinated neuron. You'll see in our diagram here in a second that the myelin sheath is not a continuous structure. Remember, it's made up of a few different cells that kind of wrap around the axon. So we have gaps in that myelin sheath, and we call these nodes of Ranvier. And so these happen about every millimeter, and the ion movement actually occurs at these nodes. So the ions kind of jump node to node to travel down the nerve. So one example of a disease that affects our myelin sheath is multiple sclerosis, or many times you'll just hear it termed MS. And so it attacks our myelin sheath, which makes our neurons much less efficient. So that can cause loss of muscle function and really loss of any function that is affected by myelinated nerve. Excuse me. So we do have some unmyelinated neurons, and they almost more like rest in a tunnel of our oligodendrocytes or Schwann cells rather than being completely wrapped and insulated. and it's typically going to be a relatively small nerve. So here on the left, we have myelinated. You see we're in the peripheral nervous system because we have Schwann cells, but it's going to wrap itself around to form this insulating layer. This is considered unmyelinated, so you still see our Schwann cells, but you see the neuron axons. These little axons are more just embedded or tucked into channels within the Schwann cell rather than insulated and wrapped. All right, so when we look at nervous tissue, it shows up kind of like two different colors because of having myelin. So gray matter is going to be unmyelinated. It's typically going to be our cell bodies and their dendrites, whereas our white matter is going to be mostly our myelinated axons, which give it that white color to it. So when you think gray matter, think cell bodies and dendrites. When you hear white matter, you should be thinking axons. So for our nerve cells to communicate, they have to be ready for the signal. So they're called what we term that nerve cells are excitable. They have the ability to be excited. So they maintain what we call a resting membrane potential. And that's kind of like its ready or set point so that when a stimulus arrives, it's ready to fire off. And so in nerve cells, this is how we communicate. And we call these nerve impulses or communications action potentials. All right, follow along with me here through the words, and then we'll walk through a diagram. And this is definitely the most important concept in this chapter in terms of the physiology. So try to stick with me here. So think of an action potential as an electrical signal that's going to travel along the cell membrane of our neurons. It's just like electricity going across a wire. So there, across our cell membrane, we have a lot of voltage-gated sodium and potassium channels. So during resting or at rest, when our resting membrane potential is maintained, our sodium potassium pump and channels are completely kind of just hanging out. They're just waiting. So when a stimulus actually gets to the nerve, what happens is, and let me backtrack for just 1 second. So let me discuss resting potential just a little bit more. So at rest, if you think back to our earlier chapter where we talked about active versus passive transport, we talked about the sodium potassium pump as one of our active transport examples. So at rest, there's a high concentration of sodium outside of our neuron or our cell membrane, and a high concentration of potassium inside of our cell membrane. So lots of sodium on the outside, lots of potassium on the inside. So keep that in mind. So when the stimulus gets there, our sodium channels are going to open for a brief moment, and that sodium that's at a high concentration on the outside of the membrane is going to rush into the cell. It's going to quickly diffuse. So the movement of that sodium into the cell is going to create a local current at that exact kind of section where the sodium is rushing in. And that is going to now change the inner part of our cell that was at a negative charge is now going to become a positive charge. So we call that depolarization. If the stimulus isn't strong enough and not enough of our sodium channels open, or for not long enough, they're just going to close again, and that potential is going to disappear locally. It's like it never happened. If we have a large enough rush or diffusion of sodium, the depolarization is going to meet what we term a threshold value, and that is going to trigger an action potential to continue to spread down the cell. So our threshold depolarization is going to cause our voltage-gated sodium channels to open. And so this is going to cause a lot more sodium to rush into the cell. So now we have a ton of sodium rushing into the cell. We went from negative charge to a very positive charge. And so eventually we're going to hit a point where that triggers our potassium channels to open. So now we have sodium coming into the cell, and we're going to have potassium starting to go out of the cell. As potassium goes out of the cell, that begins to reverse our charge. That charge reversal, as we're going back negative, is going to cause our sodium channels to close. And so now we have no more sodium coming in, potassium still going out, and that's going to result in repolarization. So we're trying to reset our initial charge. So as we reset our initial charge, we actually go a little bit too far. We go past where we need to, and we call that hyperpolarization. And then our sodium potassium pump from there will get us back to our normal resting potential. So action potentials occur all or none. So we said we have that local potential that starts it, but once that local potential meets the threshold value, there's no way to stop it. It's going to keep going. Okay, so this is we're going to walk through this diagram. And so if you have a good understanding of our action potential physiology, you should feel pretty good about being able to explain these next few slides in this graph here on your own. So at resting potential, we are negatively charged inside of our membrane. So we're at a negative 70 millivolts inside of our cell membrane. Our sodium and potassium channels are all closed. We have a high concentration of sodium outside the cell and a high concentration of potassium inside the cell. So this is our resting membrane potential. So with depolarization, we get our local potential. So we get some sodium rushing in. If that sodium that initially diffuses in changes the charge enough to go past the threshold, then we're going to get our voltage gated channels wide open and tons of sodium is going to come in. So sodium's got a positive charge, so it's going to take our negative resting potential and skyrocket it positive. That's depolarization. So once we hit depolarization, we get to a certain point that triggers our potassium channels to open. Remember, these voltage-gated potassium channels are going to allow this high concentration of potassium inside of the membrane to rush out of the membrane. So now we're losing positive ions, and so we're going to go back negative. And then this shows that hyperpolarization where we go more negative than we started, and then it's going to get restored by the sodium-potassium pump. So resting potential here at negative 70, local potential crosses the threshold, sodium rushes in through the channels, and depolarization happens to trigger an action potential. The potassium voltage-gated channels open, potassium rushes out, and we go back to negative. That's hyperpolarization. And then our sodium-potassium pump is going to restore us back to normal resting potential. So there's not much that you need to know about this slide other than the fact of remembering which goes with which here. So action potentials along unmyelinated neurons, we're going to call continuous conduction. We don't have any gaps in the myelin. It's just in that channel, so it just goes straight across. We call myelinated axons, their action potentials are known as saltatory conduction because they're jumping node to node. So unmyelinated is continuous and myelinated is saltatory. Okay, last thing for this first part here, I think. Yeah, we'll finish with the synapse. Okay, so a neuroneuronal synapse is a junction or a connection where one axon of a neuron is going to interact with another neuron. So usually it's going to interact with the dendrite. So we're going to talk through the anatomy or kind of what's going on at this junction. So our neurons have chemical substances called neurotransmitters. So even though it travels in a signal, that action potential is like electricity, and we actually want to communicate neuron to neuron, we're going to have neurotransmitters. So that action potential can trigger the release of neurotransmitters. So two common ones are acetylcholine and norepinephrine. And so the specific channel type, specific receptors, all that's going to depend on the type of neuron, like where it's located, and the type of neurotransmitter that it responds to. So the neurotransmitter is typically going to say, yes, fire more action potentials, or no, stop firing. So these neurotransmitters don't hang out indefinitely. They only have a short duration effect. And to prevent it from having a long duration effect, we have enzymes that break down the excess neurotransmitter within the cleft, and our presynaptic terminal can always reabsorb some of its neurotransmitter. So let's look at this diagram here. This is the easiest way to go through it together. Okay, so this is our nerve, this is the axon, and here is our synaptic bulb. Here's our synapse. So we have this gap here prior to interacting with the next neuron or tissue. So the action potential is going to spread down. We're going to have vesicles. These little bubbles contain our neurotransmitter. When the action potential reaches our synaptic terminal, calcium channels open up. These calcium channels cause the release of these synaptic vesicles, which allow release of our neurotransmitter in our synaptic cleft. So presynaptic terminal has the neurotransmitter. Calcium rushes in when the action potential gets there, and that causes release into the cleft. So now that the neurotransmitter is in the synaptic cleft, it can either bind with receptors or travel through channels on the postsynaptic membrane. So now you see, if we zoom in more, our neurotransmitter binds to these channels, our sodium channels open, and that's causing sodium to go into the cell. So in this case, the action potential is causing the next cell to allow sodium to enter. Okay, we're going to go ahead and stop part 1 here, and we'll hop back in for part 2 here shortly.